Okay, well, this week I'm going to talk about the kidneys, renal physiology. Primordial sea oceans of nephrology, just to take a pee. All right. I dove into this. Were there any, I should have asked first, were there any questions about anything we covered last week? No. So back to our buddy Hippocrates. 400 BC, the I guess father of Western medicine. He started out, amongst other things, as a uroscopist. A uroscopist is an individual that has you urinate into a jar and then inspects the urine to see what color it is, swishes it around, sees if there's bubbles in it, takes a whiff, what does it smell like, takes a little swig, what does it taste like, and helps diagnose disease from this. In fact, it was Hippocrates, I believe, this is correct, that was the first person to make the distinction between diabetes mellitus and diabetes insipidus. Diabetes itself is a general symptomatic description of a disorder that is accompanied with excessive urination, excessive micturition. Diabetes, the shedding of lots of fluid. And diabetes mellitus is the situation in which that urine is, mellitus is the root word like mellifluous, means sweet. A mellifluous sound is a sweet sound. Mellitus means sweet urine. There's a high level of sugar in the urine, whereas diabetes insipidus, an insipid person, is kind of a bitter person. And certainly diabetes insipidus is urine that doesn't taste quite as nice as diabetes mellitus. Our ever stubborn physiologist Galen, persistent in his desire to undermine the Greeks, dominate them with good Roman medicine, not able to experiment on humans, thankfully. The physiologist, the anatomist, the physician to the gladiators, his hypothesis was that the kidneys were what produced urine. And so he took living apes of some sort, and while they were alive, opened their backs up and clamped their ureters and watched the kidneys themselves swell and fill with urine, and thus concluded that it was the kidneys that actually produce urine. So the first person to correctly deduce the function of the kidneys. Meanwhile, Hippocrates, his tradition of uroscopy continued well into the Middle Ages. Here we see an example of someone in the tradition of Hippocrates inspecting a flask of urine in 1600. So you weren't feeling well, you had a problem, you would go to one of these uroscopists, pee in a jar, and they would somehow divine your that whatever physical or metaphysical ailment plagued you. However, this guy named Thomas Bryan finally blew the whistle on this whole endeavor in a tract that he wrote called the Piss Prophet, or certain piss pot lectures that he delivered. And I'm going to just read you the inscription here. Wherein are newly discovered the old fallacies, deceit, and juggling of the piss pot science, used by all those, whether quacks or empirics or other methodical physicians who pretend knowledge of the diseases by the urine in giving judgment of the same. So he goes on to basically tear apart this entire kind of sham profession. Do I have a picture? No. This is not to say that these uroscopists didn't make significant scientific contributions. There was one of these uroscopists around the same time named Henning Brant, who was from the Netherlands. And this guy was an alchemist and a uroscopist. And as a uroscopist, you'd go to him like this and you'd get your disease diagnosed and whatever, you pay him some coins. But he was also in search of the Sorcerer's Stone as an alchemist. An alchemist is a person, so you're probably familiar with the term from Harry Potter. The Sorcerer's Stone is this legendary material that does a couple things. It, first of all, can turn gold or lead into gold, and then it also confers immortality. So a lot of these people were in search of this. Henning Brant sort of combined his two passions here. Really strange character. He needed a lot of money to build his lab, so he married a wealthy widow, and she promptly died. And he married another that was wealthy, and she also promptly died. I can't say anything more about that. But he inherited a pile of money that he built a lab with and a couple stepsons that he sent out into the taverns and breweries of the lowlands of the Netherlands and collected an enormous amount of urine from these establishments, the alcoholics that his stepsons ran across, and filled like 1,500 gallons of this stuff into a container in his basement in the lab. So just to give you a sense, that's larger than the volume of this room. And so he let the urine sit in the basement of his house and putrefy for several months until he determined by the aroma that it was certainly ready and began to boil this stuff down. You can't begin to imagine, I certainly at least can't begin to imagine what it would smell like boiling 1,500 gallons of rank urine down by fire in the in the 17th century. So eventually he reduced 1,500 gallons down to this. Well, there were several stages that he went through, but eventually he got a tiny, like golf ball sized mass of glowing white waxy substance, like glowing in the dark, glowing. If I may give it away if I were to tell you phosphorescently glowing. He extracted elemental phosphorus. It was the first time that elemental or white phosphorus was isolated from the phosphates that would be found in human urine. He, of course, thought it was the Sorcerer's Stone, and while it wasn't that, it indeed was a major economic windfall. Not so much for him, because he wasn't a very good businessman, but for the rest of the world, particularly the soon-to-be burgeoning military-industrial complex that put white phosphorus to great use as an explosive. So matchsticks, that's something that we got from it. Anyways, uroscopy, not entirely a waste of time, I guess. We do have matches from it. Moving forward a little bit was this guy, Richard Bright. A little bit more, little less radical story. But this guy was the first person to show the connection between what's called proteinuria, or the presence of protein in your urine. And he made a three-way connection between protein in the urine, what he called dropsy, which is edema or swelling, massive swelling, as is illustrated here in this poor woman. And the third point in the triangle was kidney disease that was, of course, eventually fatal to these people. So this woman here is not long for this earth. She's in the midst of acute kidney failure. So he demonstrated this in 1827, and this was really the beginning of modern scientific nephrology. So glomerulonephritis, we'll understand the roots of that word in a bit as we go through the anatomy of the kidney here. Glomerulonephritis, or an inflammation of the functional unit in the kidney, was known as Bright's disease for a very long time after this. So a guy was in Edinburgh. Edinburgh, I don't know how much I've talked about Edinburgh, but Edinburgh was in the late 18th and through most of the 19th, even into the 20th century, one of the greatest places worldwide for anatomical and physiological study. So into the modern era, another interesting point in time is this guy here, Joseph Murray. By all accounts, a really pleasant fellow, and I can believe that just by looking at the picture of him. This was the first guy to ever successfully endeavor a kidney transplant. So the first kidney transplant was not done by him. It happened in France, and they took, I believe, a kidney from a chimpanzee, and they tried to transplant it into a human in 1909. There was tissue rejection, of course, because of the immune response, the MHC incompatibility between monkeys and humans. But in 1954, this guy got around the problem by taking a kidney out of one of the Herrick twins, these two guys here in the suits. They were identical twins, genetically identical twins, so there was going to be no tissue rejection. He took the kidney out of one, stuck it in the other successfully. You see them here also in inspecting the first real dialysis machine. Dialysis is a technique by which you remove the blood from the body and filter out any of the toxins that are in there. It's a very crude process, but also attributable to this guy, Joseph Murray, who was here in New England. The first kidney transplant happened in Boston. So, yeah, this is a picture of the actual surgery. Cool. All right, so that's the history of nephrology. Short, fast. First, the anatomy of the urinary system. The four components are the kidneys. This is the part of the kidney that actually produces urine. The ureter, which, as Galen discovered, transports urine from the kidney to the urinary bladder, which is where the urine is stored prior to its elimination from the body through the urethra. So the urinary system is primarily involved in the excretion of waste materials from the body, principally nitrogenous waste materials, although there are other things that are eliminated from the body by the kidneys, and then, of course, elimination of this fluid waste. There are other functions of the kidney, though, and we're gonna put half of our time into talking about excretion. I probably won't even talk about the micturition reflex of elimination, the urination reflex. But then the other half of this that is less appreciated, I think, by junior students in physiology is the homeostatic regulation that the kidneys are involved in. They are crucial. We already know some of this, don't we? We know about the effects of parathyroid hormone on the kidney and how the kidney is then able to turn calcidiol into calcitriol. Right, that happens at the kidney. But we're going to learn about a lot more that the kidney does. So it helps regulate the plasma volume through the hormones erythropoietin and renin. We'll see what those do. I won't talk about erythropoietin very much. Erythropoietin is responsible for the process called erythropoiesis, or the formation of new red blood cells. It stimulates the formation of new red blood cells. But we will talk about renin at length. And then it helps regulate solute concentration. For example, through the action of calcitriol, right? And we've already covered how calcitriol can affect the plasma concentration of calcium, for example. It definitely stabilizes the pH in the body by managing the hydrogen ion concentration and the bicarbonate buffering, and then it is able to detoxify the blood. So it's able to take some waste materials, toxins, not waste materials, toxins that are in the body, and solubilize them for export in the urine. All right. And, this is not really an anatomy class, but to talk, the structure function paradigm that we've talked about since the beginning of the semester requires me to go into a little bit of anatomy here. So I'm going to go over some of this. By the way, I have put the entire outline for all lectures this week on. I don't know if any of you ever look at it, but that outline has been extended for the entire week on this renal chapter now. So where should we start? Let's start just by identifying the cortex and the medulla. So the outer part of the kidney is the fibrous capsule, and then just deep to that is this renal cortex. It's this band of tissue that wraps around the kidney, and then on the inside is this renal medulla. The renal medulla is made of essentially two portions. There are the renal pyramids, that's these triangular shapes that you see here, and then in between them are renal columns. The renal columns are more or less conduits for the vasculature that is serving the kidney. The renal pyramids are where the nephrons reside, and they terminate in these renal papillas. And a renal papilla just means a small hill. The renal papillas have these perforations in them that are where the nephrons terminate in these papillary ducts. We'll see a diagram of that in a moment. But this is where the urine emerges into these minor calyces. Plural is C-A-L-I-C-E-S, calyces. The minor calyces converge into a major calx. There are about three or four major calx in a kidney. Those then again serve as tributaries to the renal pelvis. The renal pelvis emerges from the kidney through an area called the hilum. A hilum, did I introduce that term when I was talking about the lung? So a hilum is a general anatomical term that just means the service conduit into an organ. The ureter or the renal pelvis passes through the hilum. Also, all the vasculature, the renal arteries and veins are going to go through there. Any innervation that's passing in to the kidney is going to pass through the hilum. There's a hilum, for example, to the lung, which is where the primary bronchi, all the vasculature, et cetera, is going to pass into the lung. Is there anything else important there? I don't think so. It's just the makeup of the kidney. And we'll have to talk about the blood supply just a little bit. The renal artery is a direct branch off the aorta, and it breaks into segmental arteries, which basically mirror the major calyces, or the calyces in general, which then arborize to send off what are called interlobar arteries. along the renal columns. At the junction between the medulla and the cortex, they then send off these sort of anastomosing or connecting branches that are called arcuate arteries. They follow an arc along the base of the renal pyramids at that boundary. Then, coming off of that, we have these cortical radiate arteries. I have in this diagram here, I should probably thought I had changed this terminology. Sometimes you'll see interlobular artery up there because the lobules, renal lobules, are defined. The boundaries are defined by these cortical radiate arteries, but interlobular artery and interlobar artery are two very similar words, and because of that, very confusing. So, cortical radiate artery is a much more common term that you'll find in the literature. Most people don't use interlobar artery any longer. Then there's about all this stuff here in purple, afferent arterioles, glomerulus, all that. We'll have to wait till another slide to see what that looks like. We'll have to zoom in a bit. But it's all functionally significant when we think about how the kidney actually works. So a lot of blood goes through your kidneys. We've talked about how much blood went through your lungs. Well, about 1/4 or 1/5 of the cardiac output, which for a typical person at rest, is over a liter per minute, passes through your kidneys. They get a huge amount of your cardiac output, a huge amount of blood flow. All right, so these are the nephrons. This is the nephron. There's two types. The nephron itself is the functional unit of the kidney. This is where urine is made. All right. There are two types. There's the cortical nephron and the juxtamedullary nephron. They are identical in everything but proportion. All right. They have the same parts, but the proportion and spatial distribution is a little bit different. A cortical nephron is about 15% of the nephrons, and then about 85% is a juxtamedullary nephron. I believe that's right. I think I don't have that backwards. And we'll get into the parts in a moment. But you can see here how a nephron goes into this collecting duct that then a number of collecting ducts coalesce into a papillary duct that emerges through an aperture on the renal papilla and delivers what is at that point urine into the minor calx that's associated with it. Looking now at the vasculature, you'll see the arcuate artery that's passing at that boundary between the cortex and the medulla and throwing off those cortical radiate arteries. These cortical radiate arteries themselves give off these smaller branches called afferent arterioles, and then hanging off all those afferent arterioles, like little berries, are the glomerulus, this tangle of capillaries, this little knot of capillaries. We'll see what that looks like in more detail in a moment. But I want just to point out the distinction between cortical nephron and juxtamedullary nephron, in that the cortical nephrons are, the name is very descriptive for both of them. The cortical nephrons primarily reside in the cortex, and the juxtamedullary nephron, the bulk of the apparatus is at that boundary between the medulla and of the renal pyramid and the cortex, with only really what's called the nephron loop, this little loop that you see ducking down there, being in the renal pyramid. Okay, a lot of anatomy coming at you fast and furious. Everybody okay with this? No deep concepts, just a lot of terms. All right. So here are some pictures of them with the vasculature put in place. So, the reason I'm going through all this vasculature, we talked about how 20 to 25% of the blood supply goes to the kidneys. Vasculature's where it's at, right? I mean, we're getting a ton of blood into the kidneys, and these nephrons are going to be trying to filter the blood. So it's important to know the exact plumbing in here. Afferent arterial. So afferent and efferent, these words are analogous to the usage in the nervous system. Afferent is sensory, meaning stuff that's coming into the brain, and efferent is stuff that's coming out of the brain, right? And that's what it means here. Afferent arterioles carry blood into the nephron, and efferent carry it out, or actually afferent not just into the nephron, it carries it into the glomerulus, and efferent arterioles carry it out of the glomerulus. We'll see what the glomerulus is in more detail in a minute. Then from these efferent arterioles, they branch out into what are called peritubular capillaries. This complex mesh, this complex network of capillaries that fully encases all of the tubes in the nephron. Highly anastomosed. So the word anastomosis is, I guess I should put a definition if I'm going to use it, but it basically is in a vascular web. An anastomosis is a cross between branches. So like a normal tree has branches, but then like a banyan tree has branches, but then they connect at different points between branches, so you can form loops. That's what an anastomosis is like. All right, so like loops in a blood mesh or network. In the juxtamedullary nephrons, one of the structural differences between that and a cortical nephron is that nephron loop, that little, what's called the loop of Henle that descends into the pyramid. It's much longer than a cortical nephron. And the peritubular capillaries there are called the vasa recta. But they're just a type of peritubular capillaries, the vasa recta. Vasa just meaning vessel, recta meaning straight, the straight vessels of the nephron that cover the nephron loop. All right. Lots of anatomy here. Did anybody look at this before coming to class? Any of you doing that? All right. So let's see here. Here's the nephron with the functional components diagrammed with all the vasculature removed, besides the afferent and the efferent arteriole and the glomerulus. So the first thing that you'll see here is this sort of round sphere on the left here. This is called the renal corpuscle. That structure is called the renal corpuscle. And this is where we make what's called filtrate. We actually filter the blood. We'll see how that works in a bit. Within the renal corpuscle, the afferent arteriole enters the renal corpuscle and then branches into this complex knot of capillaries that we call the glomerulus. So now you can begin to see what glomerulonephritis might be. It's some kind of inflammatory damage to the nephron, specifically at the glomerulus. And we'll see how that happens in a bit, glomerulonephritis. And then the blood emerges from the renal corpuscle through the efferent arteriole. That the corpuscle is then surrounded by this capsule, this renal capsule, and there's a capsular space into which the filtrate that's coming out of the blood passes. So the renal corpuscle has two ends. There's a vascular pole. This is where the afferent and efferent arterioles enter and emerge. And then there is the urinary pole, the other end, the urinary pole. This is where what is filtrate and will soon become urine passes into a series of into a long series of ductwork, of tubular ductwork. The first part of that duct work we call the proximal convoluted tubule. Proximal meaning near to the nephron, to the renal corpuscle. The word proximal is just an anatomical term that means close to something, like to approximate something means to come close to it. The proximal convoluted tubule then ends and goes into what's called the loop of Henle, or the nephron loop. The Henle is an eponym, a name for the person that discovered it first, or described it. And this has a descending loop and an ascending loop. And I kind of like, when we get to talk about the nephron loop, it's pretty amazing how it works. So you'll have to hold your breath and wait till then. And then the nephron loop comes back up into the region of the proximal convoluted tubule, but now is called the distal convoluted tubule because it is on the far side of the nephron loop from the renal corpuscle. Distal convoluted tubule, then at the end of it, it joins with the collecting duct, and then you've already seen how it coalesces into a papillary duct for the delivery of urine to the calyx. Am I going to go through the little bits and bobs here? Yeah, I think I will. Might as well. So at the glomerulus, this is where we have filtrate produced. We'll see how that works in another slide or two here. The proximal convoluted tubule, the principal job of the proximal convoluted tubule is simply the reabsorption of water. When we have a filtrate, we actually lose a huge amount of water in that process. And we would rapidly dehydrate and die if we were not able to reabsorb the vast majority of the water that is filtered out in the glomerulus. We also begin to reabsorb a number of important ions and any of the organic nutrients. So all of the glucose that is in your blood, that is filtered out and it needs to be reclaimed, or else it would just be throwing the baby out with the bathwater, so to speak. The same with all of the ions, the important ions that are in this filtrate. Next, we go into the loop of Henle, and there's this process called countercurrent multiplication that we'll talk about. But the job of the loop of Henle, the descending loop is the resorption of water, and the ascending loop is the resorption of sodium. And then sodium chloride. It's a neutral, charge neutral resorption. And then the distal convoluted tubule, this is where we kind of fine-tune the filtrate, which looks much more like urine at this point. We've pulled most of the water out of it. We've pulled everything that we want to keep, like the sodium and the glucose and whatever, the other nutrients that we may want. But this is where, if there are any toxins in the blood that we want to actively excrete, so we're going to actively excrete toxins. Maybe, I don't know, maybe we have too many of some sort of ion in the blood. This is where we're going to adjust that. And there can be a variable resorption of water. So this is going to be a site for hormonal control. We're going to talk about various what are called axes for the control of water and electrolyte balance at the distal convoluted tubule. This can continue in the collecting duct as well, but also under hormonal control. All right, questions here. This is just an overview. So let's go through it in more detail. Here is a picture of the renal corpuscle. I don't know why it's blue and green, maybe just to like highlight different portions of it. But anyways, so what you see here is the afferent arteriole entering into this glomerulus, this tangle of capillaries. And the size of one of these things is anywhere from 150 to 250 microns in diameter. Pretty small thing. Here's a cartoon of it. So we see that the afferent arteriole brings blood in, goes into this knot of capillaries, and the blood then emerges from the efferent arteriole. What you'll notice also is that there's this glomerular capsule that has a parietal and a visceral layer, just like we talked with serous membranes. Parietal meaning the wall of the capsule. That parietal layer then wraps around and joins with the visceral layer. This visceral layer of the glomerular capsule lays right on top of the capillaries, the capillary epithelium of the glomerulus. It covers all of those capillaries. Specifically, the cells of the visceral layer are called podocytes. P-O-D-O-C-Y-T-E-S. Podo meaning foot. Podocytes. I don't know, I guess the first person that described these, and I don't know who that is, but thought they looked like feet. And these, one of the things that you notice about your foot is that you have toes on your feet. Well, these podocytes have toes as well, and we call them pedicels. We'll see. Actually, I guess I don't have them labeled here. But you can kind of see this like zigzag pattern in the drawing here. That's meant to represent pedicels. We'll see what that looks like in a moment. So anyways, the blood comes in and the fluid filters out into the capsular space and then leaves the urinary pole of the renal corpuscle through the and enters the proximal convoluted tubule. So when you look at this, the afferent arteriole is larger than the efferent arteriole, right? And at least in this drawing. Is there any reason why the artist might have done that? What is that conveying? It's conveying the anatomical reality, and why do you think that might be? Yeah. Absolutely. So there's two things here. First of all, you want to keep a pressure head against which the blood pressure can work. So it's kind of like the idea of afterload, right? In the heart. Well, maybe I shouldn't invoke that one because it's got an opposite intent there. Having the efferent arteriole be smaller, that keeps the pressure up in the glomerulus and increases the filtration. So it's kind of like any of those like backyard things that you screw into a hose that you played with as a kid, like a slip and slide or like some kind of hose with little jets of water coming out it. If you take the cap off the end and relieve the pressure, those jets go down, huh? And if you slow the current, you increase the pressure and you increase the flow of filtrate. There's another reason. There's another practical reason why this would happen. So it's related. It's a consequence of the first. We're losing volume here, aren't we? We're actually losing volume. And so there's less blood coming out of the efferent, or coming out of the renal corpuscle than going into it. All right. So let's look again. Let's continue to add a little more detail. So you can see here this time the pedicels of the podocytes. These projections, they're just like finger-like projections that are coming off the podocytes, and they interdigitate. They kind of move together like this, all right? And they act as filtration slits through which the fluid, what is the filtrate, will pass between them. It's sort of like taking your hands like this and trying to fill them with water. You're not, maybe you're going to the beach and scooping up a bunch of sand with your hands. The water is going to pass between the cracks in your fingers, but you'll be left with shells and sand in your hand. All right, that's what's going on there. Another thing you're going to notice about this is that there's this region on the vascular pole called the juxtaglomerular complex, which is made up of two parts, a macula densa and juxtaglomerular cells. So this juxtaglomerular complex is made up of part of the renal corpuscle and a region in the distal convoluted tubule. So what happens here is after it emerges from the nephron loop, the distal convoluted tubule does its thing and swings by the beginning of the whole process here. It swoops in, and these cells interact with these juxtaglomerular cells that are embedded in the tissue of the vascular pole. This is, should I tell you or should I let you guess? What do you think this would be for? Can you reason out why it may be useful to have cells from the end of this process of tweaking the contents of the filtrate come back and interact with cells at the very beginning? Are the gears spinning or grinding? Should I just say it? It's a regulatory process. It's like quality control. This is the apparatus in the nephron that enables the nephron to adjust the quality and volume of the urine by taking the filtrate at the end and comparing it to what's happening at the beginning of the process. This is how we get a feedback loop. This is the origin, the anatomical origin of the homeostatic processes that we'll talk about. So this juxtaglomerular process is essentially endocrine tissue, and we'll see how it works in a bit. Some other things to point out. So over here, you see a cross-section of some of the capillaries in the glomerulus. And between these adjacent capillaries, we have this cell called mesangial cells. And they are contractile cells that help control the diameter of the capillaries in the glomerulus, and in so doing, they change the capillary blood flow. All right. Is there anything else to say about this slide? I think not. Okay. So let's get into how this works. We've got all the anatomy in place that we need to talk about all of this, which is good. That's what I wanted to get today. So glomerular filtration. It is a passive process. We don't actually burn ATP to make this happen. I guess we do, in a sense, in terms of having our heart drive our blood and create blood pressure, but it's really the blood pressure that drives the filtration. So blood comes in through the afferent arteriole, and in this bottom picture we see the blood inside the capillary in the pink region, and then fluid passes through the filtration slits and various small solute particles. Proteins are rejected, and any kind of large blood cells, any large solute is going to be excluded and not able to pass through these filtration slits. But small water-soluble molecules will be able to pass through. So glucose, for example, and amino acids, and urea, and ammonia, and et cetera, things like that. If we look even more closely at this membrane, this glomerular filtration membrane, you'll see that the epithelium that forms the capillaries, it's called fenestrated. It's fenestrated epithelium. It has these small perforations or little holes. Fenestra is the French word for window. I guess it must come from the Latin. There are these little holes or windows in the cell that allow the fluid and the small solutes to pass. And then on top of that are the podocytes with their filtration slits made between the gaps between adjacent pedicels. All right. So the one last thing to point out, this actual filtration pressure is only 10 millimeters of mercury. It's only about 10 millimeters of mercury. Does that seem suspicious to anybody? I told you that blood pressure is driving this, right? What's our blood pressure, more or less? Let's say, what's a normal blood pressure? About. It's like, we just finished that. Yeah, 120 over 80 millimeters of mercury. So the mean arterial pressure of 120 over 80 is something like what? Like 100 or something like that? 95 millimeters of mercury. But we have 10 millimeters of mercury filtration pressure. Huh, what's going on with that? Well, we'll get to that. Can have to wait maybe till next time, but we'll see here. Anyways, this is a micrograph just so you can actually get a sense now of what this membrane looks like. This is a podocyte sitting on top of the glomerulus, this one cell, and it has all these little fingers that branch off of it. All right, there are these little projections that form the filtration slits. Pretty cool. Early SEM of it. I don't need this. I made this slide visible, but I should have kept it invisible. All right, so here's the answer to my question. I think I can just get this out before I have to let you go. So the blood hydrostatic pressure that's coming in is about 60 millimeters of mercury due to the aperture in the capillary and blood pressure drop as it comes in. About 60 millimeters of mercury blood pressure, pushing solutes out of the blood into the capsular space. But there are two forces working in opposition to that. There's a colloid osmotic pressure. So colloids are things like the proteins, the large solutes that are not able to pass through the filtration slit. All right. There's this osmotic pressure between, just like a dialysis membrane, if for any of you who've maybe been in a biochem lab and used a dialysis membrane. But you have a high concentration of these proteins on one side in the blood and none in the filtrate. That has a reverse osmotic pressure of about 30 millimeters of mercury. And then there's also the back pressure of the capsular space itself. There is a hydrostatic pressure in the capsular space. That's about 20 millimeters of mercury. So there's about a net of 10 millimeters of mercury pushing filtrate out of the glomerulus. The problem here is if you have high blood pressure in the glomerulus, it can break this structure down. It can break the structure down, and because of that, the glomerulus is really susceptible to high blood pressure. You can rupture these glomerular capillaries. It leads to scarring and damage of it, nephrosclerosis, atherosclerosis of the renal blood vessels, and can lead to renal failure. It can lead to the destruction of the glomerulus. That is called Bright's disease, the glomerular nephritis. So high blood pressure is probably what happened to that woman who had the edema in her legs, the dropsy, glomerular nephritis. Okay, that's it. Thank you.